Okay, well, thank you for the introduction. As Mark mentioned, I'm the technical sales specialist for APAC, and I'm located in Brisbane, Australia. And today and I specifically look after our genomics digital spatial profiler. So I'll be talking about that instrument today. I'll introduce the technology and give you a case example of how we use it. So Alex, he provided us with an actual great a practical example of the evolution of genomics research and how it's looking now. But if we consider the evolution of genomics research, so bulk methodologies, including NGS and gene expression profiling, have provided important discoveries in both health and disease. However, a limitation of bulk methods is the homogenization of tissues. And when you homogenize tissue, you're losing cellular granularity in terms of which genetic signatures are generated from specific cell types. And that's represented by the fruit smoothie here on the left. So if we fast forward to the advent of single-cell RNA-seq, researchers are now able to use a multi-omics approach to phenotypically profile and identify novel cell types on the molecular level. And again, Alex provided a really good example of some work that's being done in that space. And so, being able to identify novel cell types is the fruit in the smoothie. And as we know, single-cell work has underpinned global atlasing initiatives like the Human Cell Atlas, also Alex's Breast Cancer Atlas Initiative. And they're attempting to identify and understand cell types in different organisms and model systems. And while single-cell RNA-seq is integral to this process, it also has its limitations. So when we dissociate tissues, we lose the spatial context. And how cells are arranged in tissue directly governs their behavior. And that's with respect to how they communicate and how they function in both healthy and disease states. So spatial context allows us to interpret these heterogeneous cellular ecosystems and build on the knowledge that we glean from these bulk and single-cell methodologies. So if we consider the tumor ecosystem, we know that it's comprised of an amalgam of different cell types. So that includes, your different immune cell subtypes. It includes your cancer-associated fibroblasts, stem cells, etc. And translational discovery and clinical oncologists are asking certain questions about this ecosystem and how these cells are communicating, how they're interacting, and how that contributes to the progression of disease. So understanding the tumor and the tumor microenvironment in this particular space is very important. So some of the questions that are being asked are, why do some patients respond to treatments and some others do not? So we've seen examples of PD-1, L1 checkpoint blockades. What is the reason for that? Can we define that using spatial biology? What is the mechanism of disease progression? In terms of biomarker discovery, can we identify specific signatures in certain compartments within a tissue to design new therapeutic targets that might address the pathology that we're seeing? And then also, what is the heterogeny and the immune response to disease in these tissues? So one of the challenges that is faced by researchers and clinicians is that they have to choose between either high expression profiling or low plex imaging platforms up until a few years ago. So on the high plex side, that's your single cell RNA-seq, your flow cytometry, NGS, etc. And they have a high degree of plex, quantitation, and precision, but of course they lack the spatial context. On the imaging side, obviously there's a low plex ability with most imaging platforms. They do have spatial context, but they lack that quantitation and precision. So what we've been able to do with the digital spatial profiler is actually leverage our knowledge and bulk expression profiling with our nCounter system and marry that with an imaging platform that can now do high parameter analysis of both protein and RNA from a single slide. So of course, another challenge that we face out of the clinic and as researchers is that we have precious samples, and that was also mentioned in a few talks earlier. So with these precious samples, if you're trying to run multi-parameter analysis, oftentimes, depending on the modalities that you're using, you have to take several slices of that particular tissue, and you may not be able to recover that particular sample. So with the DSP instrument, we're able to take a single sample and analyze several analytes. So the types of samples that we can run on our instruments, anything that you get out of the clinic obviously is varied, and also, from preclinical models as well. So we can do core needle biopsies, FFPE tissue blocks, fresh frozen tissues, and we can also do tissue microarrays, which I think is a very unique feature of what we can do. And so how do we accomplish that? So on our platform, we use two types of reagents. We have imaging reagents and profiling reagents. On the imaging reagent side, you'll also hear us use a term called morphology markers. So we have a 4-channel microscope on the instrument that takes an image of the tissue that's on your sample. And our platform is open, But what you can do with it, as long as you have a monoclonal antibody that's conjugated to a fluor that excites in the FITC Cy5, Cy3 or Texas Red channel, we can image that on our platform. We also have a collaboration with ACD Bio, and you can use RNA scope probes to identify targets on a molecular level to identify tissues that are important to you. So what you do with these morphology markers, you bathe your sample in those morphology markers or imaging reagents. As I mentioned, we take an image of that tissue sample, and if you can look at this cartoon over here on the right, you can scan over that fluorescently labeled image on our software, and you identify regions of interest that are important for the biological question that you want to answer. And then out of these regions of interest, we're able to collect content out of that, and that leads us to our profiling reagents. So in terms of our profiling reagents, how they work, as I mentioned, we both have protein and RNA content. So on the protein side, we have monoclonal antibodies, and on the RNA side, we have in situ hybridization probes that sit on the back of that RNA transcript. So we don't do any kind of poly-A capture. We have 5 to 8 probes that sit on the most stable region of an mRNA target. And what we do is we quantitate according to these DNA oligos. And these DNA oligos are covalently linked to the antibody and or the FISH probe via this UV photocleavable linker. And so when we identify, if you look in the second panel here, when we identify a region of interest that we want to profile content out of, we shine UV light over that area. Those barcodes are then cleaved off of the antibody or the ISH probe, and then we collect those barcodes into the instrument, and then we run downstream preparations to run them on our nCounter platform or on an NGS library. And on the right-hand panel, what you can see here is a fluorescent image of a tissue. You have all these circles here labeled from 1 to 11. These are our regions of interest. We shine light through those particular areas, collect barcodes, and then we have a variety of analysis tools in our software. So those are comprised of heat maps, volcano plots, etc. So this is a high-level overview of that particular workflow. Again, you stain your sample with an imaging reagent, so that's fluorescently labeled antibodies or RNA scope probes. You identify your regions of interest on the tissue. We cleave those barcodes off the tissue. They're sipped up into the instrument and deposited into a 96-well plate. And then they're either run through a hybridization protocol or an NGS library prep to run either on the nCounter or the NGS systems. So another unique feature of our technology is that we have something called a digital micromirror chip. There's 2 of these. They're about the size of a thumbnail. And what's unique about them is they can be configured in different configurations to outline non-contiguous regions of the tissue. So if we look at the example here in the middle, this is a tumor tissue section. It's stained with 2 different morphology markers. And yellow here is pan-CK to outline the tumor, and the magenta or pink sort of color is the nucleic acid stain. And what the instrument can do is we can apply a binary mask to that tissue section. And so on the top here, we have a tumor mask, everything here in white. So we shine UV light through that area, and then we collect content, RNA or protein, out of the tumor. We can flip that binary mask over, and then we can sample out of the tumor microenvironment. And we also have an increased degree of flexibility in terms of the region of interest selection and the profiling strategies that we can use. So your first port of call is a geometric design, so circles, squares, or freehand drawn polygons, as I mentioned before. We can run a segmentation strategy, so that's similar to the example I just gave, or a tumor microenvironment. You can outline rare cell types and populations within this region of interest as well. In this example, CD3 cells. You can run a contour profiling strategy. So you can lay these contours over, say, the tumor here on the left-hand side along the invasive margin. You can look at tumor infiltrating lymphocytes, cytokine signaling, apoptotic pathways, etc. And or if you want to use an unbiased approach, you can grid along the entire tissue section and sample content out of that. In terms of the ROI size, you can go down from anywhere from 50 microns up to 760 microns in diameter per circle. In terms of some analysis tools and on pathway analysis on the top and left side here, this is called the joy plot. So you can put your region of interest here in the middle. The dials here along the outside and correspond to particular pathways. And then the spikes in the middle correspond to particular genes that are highlighted from any particular morphology marker. We also have reactome plots that are highly interactive. On the biomarker discovery side, here's a contour plot with the tumor here and tumor infiltrating lymphocytes and immune cells on the outside. We can segment these compartments and then look at gene enrichment profiles in each of these different sections. We also recently just released a single cell deconvolution algorithm. So what you can do, similar to what Alex mentioned before, is you can either input genetic signatures for cell populations from a public database, or integrate your own single-cell RNA-seq data, and then you can look at the abundance of those expression patterns across the tissue section that you're profiling. In terms of the content that we have available, so this was just released at the beginning of August. This is called our Cancer Transcriptome Atlas. This encompasses 1,835 genes across 112 pathways. In terms of content, it covers tumor biology, the immune response, microenvironment as well. It's heavily curated annotations, and it's built off the back of our success in the cancer space on our encounter profiling. It's essentially what we believe cancer is, and this is work we've done and designed in concert with oncologists and cancer researchers. You can customize this project or product and add up to 60 different targets, and the sequencing requirements this are 30 to 50 million reads per sample. Also available through our technology access program, I'll discuss that a little bit later, is our whole transcriptome atlas. This is over 18,000 genes, and this is going to be available in October, so in just a month's time. And this is highly curated as well. So we went through the literature and we removed the top 10 high expressors, mostly beta tubulins, mitochondrial genes, ribosomal genes that aren't of interest to our researchers. You can spike them back in if you want to, but that's a way to save on sequencing dollars. This product is also customizable, and the sequencing requirements are 100 million to 150 million reads per sample. So in terms of what instruments that you can run those particular libraries on, here's a fleet of Illumina instruments. And as you can see, the Cancer Transcriptome Atlas, our whole transcriptome, we're also going to have NGS-enabled protein content early next year. It sits very nicely with the NextSeq 550, but also very nice for the NextSeq 2000, which Illumina just recently released. Also just give you a brief slide on the protein side of things. So protein is read off of our encounter platform. We have the most protein content available on the market. And how these work is that you'll select a human protein core. This is an immune cell profiling core, and this has housekeeping genes for normalization attached to it. In the center of it, this is a 20-plex core. And then from that core, you can build out in 10-plex modules. So you can plex up to 96 targets on the protein side of things. We also have content for neuroscience and also for non-clinical models for mice as well. We can also do a very small 84-plex immune pathway that's cancer-related on our encounter platform as well. Oops, excuse me. And so our design as well, it encourages integration of metadata. So, Alex, again, I'll use him as an example. I mentioned single-cell RNA-seq data. You can incorporate bulk expression profiling. You can also use H&E stained and RNA scope stained slides to use as to help select your region of interest. And you can integrate all of this data and do comprehensive analysis on those. We see several examples of this in the literature. So in terms of publications, we have 26 peer-reviewed publications to date. We just published another one from the Broad Institute earlier this week. And last week in preprint, we had our first paper from a COVID-19 study that was done from Massachusetts General Hospital. And so we're publishing in high-impact journals. A lot of these are hitting the cover. And we had back-to-back papers in Nature in January. And one of the more attractive features as well is that we have research groups that are publishing within 7 to 10 months of their first paper. So I'll just give a final case study here to close up. This is one of the first scientists to make use of our system. His name's Dr. David Rim from Yale University. He's heavily focused in pathology. And he's published 3 papers using the genomics system. And here I'd like to just briefly introduce one study that was released earlier this year in clinical cancer research. So just a bit of background, in advanced stages of non-cell lung cancer, about 15% of these cases, patients regularly being treated, or these patients are regularly being treated with a PD-1 checkpoint blockade. And the overall survival in these groups after 5 years plateaus to around 40%. And because a large number of these, excuse me. There you go, apologies. And because a large number of these patients don't respond to treatment, the discovery of new biomarkers could potentially better identify patients who would benefit from treatment. So what this group does, or what they have done in previous papers, is they've stratified segments across the tumor to identify specific signatures in specific compartments. So what they did is they had TMA samples, which they segmented, and then they looked at the expression of 44 proteins across 4 different tissue compartments. So that was comprised of the tumor, a macrophage compartment, a total immune compartment, and a non-immune stroma. And then they looked at the expression of those proteins in each of those particular compartments. So here was their ROI selection strategy, and just gives you an example of what we can do with the instrument. So pan-CK here in green, staining the tumor. CD45 is an immune stain. CD68 to stain macrophages, and they ran through a segmentation profile. So the tumor here is outlined in green, CD45 compartment in yellow, CD68 macrophage compartment in the light blue, and then they also looked at that non-immune cell stroma, which is identified here in purple. So here are some of the main results of the study. They identified 2 potential biomarkers that corresponded significantly with both progression-free survival and overall survival, and you can see on the top panel there, CD56 and CD4, and this was noted in the CD45 segmented compartment. So what I've not shown on the slide, and I apologize for not having the figures, is that high levels of the proteins VISTA and CD127 in the tumor compartment were associated with immunotherapy resistance. So this is an important finding that can help identify patients who will not benefit from the PD-1 checkpoint blockade. So this is important guidance or potential guidance for therapy decisions. So both CD56 and CD4 were only found to be significantly linked to survival in the CD45 enriched compartment of the tissue, as I said before. And when studying the whole tissue sample or the tumor part of the tissue alone, the expression of these markers did not show a significant connection to patient survival. So this circles back to the importance of spatial context, where in bulk analysis, you wouldn't have been able to identify that particular signature. So this is my final slide here. I mentioned our technology access program. So if you are interested in trialing the technology, you can contact us. So the TAP program was enabled for customers to rapidly evaluate our technology to see before they wanted to go on and maybe use an instrument of their own. So what we do is we collect an FFP sample from you. You can send fresh frozen tissues as well. And then we work in concert with myself and our TAP scientist in Seattle to help design your experiment. And at the end of it, we give you a comprehensive report. If you're interested in running large-scale projects, we have a service provider here in Australia that's being run out of Griffith University, and please contact me in my email address here below if you're interested in accessing any of these services. And that's the last one I have. So thanks very much for listening to me. I appreciate it.